Our today's topic is muscle tissue, and there are three types of muscle tissue. As you can see, here is a microscopic image of the three types of muscle tissue. This is skeletal muscle tissue, this is cardiac muscle tissue, and this is smooth muscle tissue. By the way, this about this muscle tissue picture is to show you that this is representing skeletal muscle tissue. This also represents cardiac muscle tissue, which is found only in our heart. And smooth muscle can be found in organs of the digestive system, in the walls of blood vessels, and in several internal organs. For example, in the wall of the uterus, the reproductive system, in the tubes of the urinary system. So look how a single smooth muscle tissue looks like. A single smooth muscle cell is shown here. This is a tissue, meaning several cells, whereas this is one cell. Do you notice that it looks like a spindle? The shape of it, I mean, with single nucleus. Here is a skeletal muscle cell which is cylinder-like and has several nuclei. Single cell of a skeletal muscle consisting of several nuclei. The reason why a single cell of a skeletal muscle consists of multiple nuclei is because it is initially during pregnancy of a baby is formed. Single skeletal muscle cell is formed by the fusion, by the union of several myoblasts, where each of those myoblasts bring with them their own nucleus, making a skeletal muscle cell with multiple nuclei. That's why it ends up to have multiple nuclei. By the way, in this skeletal muscle tissue, this is a nucleus, this is a nucleus, this is a nucleus, this is a nucleus, this is a nucleus. And in the case of cardiac muscle, which is found only in our heart, do you notice that it is branched? By the way, this is a junction between several cells. What we see here is a single cardiac muscle cell. Notice the branching appearance. I also want you to notice these lines. They are important. These lines. They are known as intercalated disc. They are the borders between adjacent skeletal muscle cells. So that means here is one skeletal muscle cell, there is another one, and the border between them is intercalated disc. This intercalated disc not only connects to cardiac muscle cells, but also it facilitates communication between these adjacent or neighboring cardiac muscle cells. Here is a nucleus, a nucleus, a nucleus, a nucleus. Usually, cardiac muscle cell has one nucleus per cell, but rarely there can be more than one nucleus in one cell. In the case of smooth muscle cell, only one nucleus per cell. So, what's the difference between these three types of uh, muscle tissues? Do you notice that the skeletal muscle cell tissue has striped appearance? Do you see the lines, tiny lines? On a magnified picture, these lines will be really visible and prominent. This striped appearance is due to the special structure of skeletal muscle. We will see that later on. You will see why a skeletal muscle tissue has such striped appearance. By the way, cardiac muscle tissue has also striped appearance, but it is not as visible as the skeletal muscle one, but those stripes are also in here. They are results of structure which we are going to study later. So, how do we differentiate one of them from the other? We can tell that this is cardiac muscle tissue by the intercalated disc. Do you see those intercalated discs? Or by the branched appearance. Do you see that this one has 1, 2 branch, right? Here is 1, then it's branching here, if you can notice. Or here, do you see a branching here? Or the intercalated discs. The intercalated discs are only in cardiac muscle tissue. You don't find them anywhere else. How about here? The striped appearance. The very visible striped appearance is a feature of skeletal muscle tissue. Whereas here you don't see neither striped appearance nor the intercalated disc, so you can tell. So these are the 3 types of tissues. Let's see some features which all of these share in common. All of these have the following features in common. Let's say these muscles have contractility, meaning they are capable of contracting. All muscle tissues are contractile, meaning they can contract. They are also extensible. They are also elastic, meaning they recoil to their original size after contracting. They are excitable, meaning they can be stimulated and get excited and generate contraction. So what are the common features of all 3 types of muscle tissue? They can be excited, meaning stimulated and made to contract, excitability. They can contract, they can extend or relax, and elasticity, meaning they can recoil to their original size after contracting. What are the functions of muscle in general? Muscle produces movement, be it in our skeleton, in our extremities, or the wave-like movements along our digestive system, or the movements are along our blood vessels. They do cause movement. The heart also pumps blood, right? That's also movement. They maintain posture. We can stand upright because our muscles have tone, which keeps our joints balanced. That's why we can stand upright with our joints in position. So that means they can stabilize our joints. It's muscles which stabilize our joints. Otherwise, we wouldn't be able to stand up because our knee joint would have flexed or our hip joint would have flexed. We can stand upright because of muscles. Generate heat. Have you ever shivered when you feel cold? During shivering, what is happening is muscles are, especially skeletal muscles, are contracting and relaxing. So that's how they generate heat. During contraction, relaxation, muscle can generate heat and warm us up when we feel cold. Let's move on to the structure of skeletal muscle tissue. By the way, from now on, we are going to focus on skeletal muscle. Does this remind you the meat you would see in a butcher shop? So look, muscle, by the way, here's the femoral bone, and let's assume this is a muscle from attached to the femoral bone, the greater trochanter here. So notice that this muscle is covered on the outside by connective tissue known as epimysium. The outermost cover around a muscle, which is connective tissue, is known as epimysium. So muscle is compartmentalized. Look at these compartments. Look at this. One compartment, second compartment, a third compartment, right? So one of the compartments is telescoped out here, and this compartment is known as fascicle. By the way, this is a collection of skeletal muscle cells. One muscle cell, a second muscle cell, a third, a fourth, and so on, right? So a collection of skeletal muscle cells covered or wrapped by connective tissue known as perimysium is known as fascicle. By the way, this was epimysium, the covering, the wrapping around connective tissue. Epimysium here, perimysium here. What does perimysium wrap? It wraps a bunch or a collection of skeletal muscle cells. So what do we have here? One skeletal muscle cell telescoped out. This skeletal muscle cell, from now on we will call it muscle fiber. By the way, muscle cell, skeletal muscle cell, and muscle fiber are synonyms. So from now on, instead of saying skeletal muscle cell, we'll be saying muscle fiber. So understand that as skeletal muscle cell. So it is also wrapped by or covered by a connective tissue known as endomysium. Where is endomysium here? Look here, look here, look here, look here, endomysium. But under the endomysium, there is additional membrane, which is the cytoplasmic membrane or plasma membrane of the skeletal muscle cell, which is known as sarcolemma. So endomysium is on top of the sarcolemma. Sarcolemma actually is plasma membrane of a skeletal muscle cell. By the way, from now on, when you see the root word sarco used, it has to do with skeletal muscle cell. Do you remember we were using the root word os when we refer to anything which has to do with bone? Similarly here, if you see the root word sarco, it has to do with skeletal muscle. So sarcolemma is the plasma membrane of a skeletal muscle cell. So what we have here is a single muscle cell. So if we go in a reverse direction, this is one skeletal muscle cell or a muscle fiber, and here are several skeletal muscle cells. This single skeletal muscle cell has, like any cell, plasma membrane known as sarcolemma, on top of which it is covered with connective tissue known as endomysium. So several skeletal muscle cells or skeletal muscle fibers, they are wrapped together in a group known as fascicle. They are wrapped by perimysium, and as you can see, multitude of fascicles come together wrapped by epimysium. They make a muscle. So this is to show you the structure of a muscle. By the way, this is showing a cross-sectional view. Do you see? This small square is magnified here. So do you see perimysium here, right? Endomysium in there, right? So, and on the outside epimysium. So they are showing us this. This is epimysium here, showing us epimysium. So notice that. So now, if we take this skeletal muscle fiber and further study it, we will get the following picture. If we take this one skeletal muscle fiber and then further examine it, we will see the following. Let's follow it here. Look, one skeletal muscle cell. Here is one skeletal muscle cell. By the way, this picture is to show you the striped appearance of skeletal muscle cell. By the way, this striped appearance of skeletal muscle tissue or skeletal muscle cell is described as striated. The word striated is used to mean striped appearance. You see the striped appearance through the sarcolemma. By the way, this is sarcolemma, which is peeled off and folded here, so you notice it's sarcolemma. Sarcolemma is plasma membrane of a single skeletal muscle cell. What we have here is single skeletal muscle cell. Single skeletal muscle cell in its turn consists of these stick-like structures, a bunch of them in one cell, known as myofibrils. So skeletal muscle cell consists of myofibrils. If we magnify this myofibril, this is what it would look like. Do you notice that this skeletal muscle cell, being one cell, it has more than one nucleus? Earlier, I said that the reason why a single skeletal muscle cell may have more than one nucleus is because during pregnancy or during intrauterine development, when a conceptus in the mother's womb grows, skeletal muscle cells are formed by the fusion, by the union of cells known as myoblasts. Those myoblasts, multiple myoblasts, come together and unite to make a skeletal muscle fiber or a skeletal muscle cell. So that union, those myoblasts which come together, bring their own nucleus. So the muscle fiber or skeletal muscle cell, which is formed by the union of those myoblasts, ends up to have multiple nuclei. That's the explanation why one skeletal muscle cell may have more than one nucleus. Let's have a look at one myofibril. Look at this myofibril. You can consider the myofibril as organelle of a skeletal muscle cell or a skeletal of a muscle fiber. I have said that skeletal muscle cell and muscle fiber can be used interchangeably as synonyms. So a skeletal muscle cell consists of these stick-like structures known as myofibrils. We have a myofibril here. Look at a myofibril. Do you notice that myofibril consists of here a bunch of thick lines and thin lines here? So this lighter part alternating with this darker part is what causes this striped appearance of skeletal muscle cell. We will come to the detail of this darker part, lighter part, darker part. By the way, we said skeletal muscle tissue consists of skeletal muscle cells, right? And skeletal muscle cell in its turn consists of myofibrils. And this myofibril in its turn consists of sarcomeres. Here is a sarcomere. We will see a bigger picture of sarcomere, but I want you to see that if you notice here, this sarcomere from here to here is one sarcomere. Look at this, from here to here. That means from Z disc to Z disc. We will see that this more on a bigger picture. Just on this one, I want you to see the sequence. That this myofibril is one of the sticks here, and this stick consists of these ones, sarcomeres. Sarcomere is structural unit of a skeletal muscle cell. This whole thing is sarcomere. We are going to study this sarcomere in detail. Muscle fiber, this whole thing, or skeletal muscle cell, consisting of myofibrils. When we take one myofibril and magnify this stick-like structure, this is what it is. And from here, from Z disc to Z disc, consists of, from Z disc to Z disc is a sarcomere. By the way, myofibril consists of several sarcomeres attached end to end. So we can say myofibril is a chain of sarcomeres. What is sarcomere? Sarcomere is structural and functional unit of skeletal muscle cell. I repeat, sarcomere is mainly functional unit of skeletal muscle cell. We are going to study this sarcomere in detail. First, let me show you on another bigger picture the structure of myofibril in relation to here is a myofibril, bigger image of a myofibril here. Look at this myofibril, that stick-like structure which we took out from the skeletal muscle cell or from the muscle fiber. I was talking about sarcomere, which is from Z disc here to this Z disc. We will have a picture of a sarcomere. By the way, from this Z disc to the next Z disc is another sarcomere. From this Z disc to the next Z disc is a sarcomere. So myofibril is a chain of sarcomeres. So myofibril consists of sarcomeres. Sarcomere is from Z disc to Z disc, this interval. We will have another sarcomere here if we had continuous. This is just cut out. So myofibril is a long chain of sarcomeres attached end to end. Okay, this was to show you a bigger appearance of a myofibril. Let's get a bigger picture of sarcomere. We are going to spend time on sarcomere. Let's see, is this a good sarcomere? Yes. Here is a sarcomere. Did I say that sarcomere is from Z disc to Z disc? Yes. What does a sarcomere consist of? Sarcomere consists of this thick myofilament and this thin myofilament. By the way, in your textbook you might see this type thick myofilament described as myosin. The reason why they use the word myosin to describe this thick myofilament is because this thick myofilament is composed of the protein myosin, and this thin myofilament consists of the protein actin, so sometimes they may describe it as actin. So sarcomere consists of interlaced, can you see in my hand? Consists of interlaced thin and thick myofilaments like this. Do you notice that they are interlaced like this? So all thick myofilaments are attached to each other by this M line, whereas the thin myofilaments are connected to each other by this Z disc. So notice that Z disc connects all thin myofilaments, whereas all thick myofilaments are connected by the M line. So in a sarcomere, M line is in the middle and Z disc is at the ends. In your textbook, they may call the Z disc as just Z line. They are the same. They are the same. Do you notice that this one is thin? So that's why they call it thin filament. This is thicker, so they call it thick filament. So why are we studying this detailed picture of a muscle cell or a sarcomere? Because literally what we are going to study is how a muscle contracts. A muscle contracts by sliding like this and relax by sliding like this. So if we assume my fingers of my right hand represent, let's say, thin myofilaments, and my fingers on my left hand represent thick myofilaments, and they are interlaced like this as you see on the sarcomere. So contraction takes place when they slide past each other, and relaxation takes place when they slide like this. So relaxation, contraction, relaxation, contraction. So we will come across a theory which says sliding principle of muscle contraction. We will see that. So now first let's study in detail the anatomy of a sarcomere. I may get another picture to show you some details of a sarcomere. Maybe let's see this one. This is the one we had. How about here? This is thinner. Let's go here. Okay, here. Okay, on this one, I want you to notice that the part of the sarcomere where you see only the thin filaments, which is this part, the lighter part is known as I band. Notice that. There is a reason why we study this. Look, lighter part. Everywhere you see the lighter part, where thick filaments are absent, where only thin filaments are visible with the Z disc or Z line, is known as I band. Whereas there where you see the thick filaments or the overlap is known as A band. How will you remember this? Easy way to remember is, do you think letter I is skinny or letter A? To me, letter I looks skinnier than letter A. So there where you have thin filaments only, you call it I band, in which the letter I is skinny, whereas the more chubby letter A is used to name the part of the sarcomere which consists of thick filaments. And you already have noticed the Z disc and the Z line, Z disc or Z line, and M line is this one. By the way, part of the sarcomere where you have only thick filaments without overlap. By the way, A band consists of both the parts where there is overlap and where there is no overlap. This by the side of the M line, where there is no overlap, is also known as H zone. So when you study, I want you to notice that what H zone is, what I band is, what A band is, what Z disc is, what M line is. You need to really pay attention to that, because it will help us to explain how muscle contracts. It will help us to understand how muscle contracts. What did we see so far? What we saw so far is the structure of a muscle. How did we start? I'm just going to repeat so you see. We saw a muscle which consists of compartments which are known as fascicles, one of them telescoped here. And then this fascicle, which is a collection of skeletal muscle cells or muscle fiber, is shown here, and then one skeletal muscle cell or skeletal muscle fiber. And this skeletal muscle cell, when taken out, and when we see it here in this picture, it consists of myofibrils, these stick-like structures. So a myofibril in its turn is composed of sarcomeres. Sarcomere is structural and functional unit of muscle cell. Actually, functional unit. What do we mean when we say functional unit? Functional unit is part of any structure in which the function of that unit, that structure, is happening. So the function of a muscle is contraction. So if contraction is happening at the level of the sarcomere, we can call it functional unit, where the function of a muscle, which is contraction, happens. Next, we saw the details of a myofibril with a sarcomere within it and parts of a sarcomere, right? We also saw thick myofilament, thin myofilament, right? In this one, thin myofilament, thick myofilament. We are going to see the details of each filament, the thick and the thin filament, probably in this picture. Look, they took out the thick myofilament from here. Do you see this rectangle? When magnified, this is how it looks like. So I want you to notice that this is myosin head. It looks like seed of some kind of plant, right? This is myosin head. Look how the thin myofilament looks like. The thin myofilament looks like a chain of beads, right? Do you see these beads? A chain of those beads, and with yellowish line wrapping around it. By the way, this chain of beads with the dimple on them, this dimple is active site, to which during muscle contraction this myosin head comes and attaches itself. We will see this myosin head, this seed-like structure, which is known as myosin head of the thick myofilament, will be attaching itself to these dimple-like structures. These dimple-like part of this bead-like structure is known as active site. So during muscle contraction, this bead-like structure or head of the myosin or myosin head attaches itself to the dimples, which are known as active sites on the thin myofilament. So notice that this thin myofilament is a chain of these beads. They are actins actually. They have active site on them. That's to which this myosin head attaches. But these active sites are covered. When a muscle is relaxed, they are covered by troponin tropomyosin chain. Look, this yellow line going over those dimples is covering them when a muscle is relaxed. When a muscle contracts, this cover moves itself and exposes the active site for the myosin head to be attached to it. We will see how contraction happens. We will see the stages, the steps of muscle contraction. There you will see this troponin tropomyosin chain moving off the active sites, exposing them, making them available for this myosin head to be attached to them. By the way, this troponin tropomyosin cover moves off the active sites when calcium comes and attaches to them. We will see during muscle contraction the role of calcium. Calcium is very important. Do you remember we studied calcium is stored in bones, and we were saying that the amount of calcium in our bone is very important. And do you remember the hormones which regulate the amount of calcium in our blood? It's very important. Without calcium, muscle contraction cannot happen. Neither skeletal muscle nor cardiac muscle nor smooth muscle can contract without calcium. Calcium is important. That's why it needs to be within normal range in our blood, and that's why it was being regulated. I hope you remember that we studied that when we studied bone. Okay, next let's have a look at a closer look at a skeletal muscle cell. When I showed you earlier a skeletal muscle cell, do you remember with the sarcolemma? Here the sarcolemma is here. Sarcolemma is simply plasma membrane of a skeletal muscle cell. And we have seen myofibrils. What we are looking here is, if we take several myofibrils, these several stick-like structures, and expose them here, how do they look like? What else is relevant to muscle contraction? By the way, I was telling you that these myofibrils can be considered as organelles of the skeletal muscle cell, right? So we have here, in addition to the myofibrils, other additional organelles like mitochondria. Do you see mitochondria here? Do you see the sarcoplasmic reticulum? By the way, do you see a nucleus here? Those are organelles like any other cell. There are special structures which you don't find in other cells. For example, look at this tube-like structure. It's known as transverse tubule. You may not find it in other cells, but we have it here in skeletal muscle cell. By the way, this is part of a skeletal muscle cell where only several myofibrils are magnified here. What do we have here? We have mitochondria, sarcoplasmic reticulum, and T-tubule. Why are we showing here? Because they have role in muscle contraction. After studying the structure of skeletal muscle tissue, skeletal muscle cell, and its parts like the myofibril, the sarcomere, the next thing we are going to do is to understand how does muscle contraction and relaxation take place. That's what we are going to learn next. So these structures have role in muscle contraction and relaxation. That's why we study them. By the way, here is also a tube which is part of this sarcoplasmic reticulum. This tube, which is by the side of the transverse tubule, the white one is transverse tubule. This one is known as terminal cisternae. It's part of the sarcoplasmic reticulum. They differ in color. This one terminal cisternae, another terminal cisternae, and a transverse tubule in the middle is known as triad. To mean triple. This is triad, triple. 3 tubes. They have role in muscle contraction. By the way, for muscle to contract, electrical impulse which comes from the nervous system, which is practically order from the nervous system for the muscle to contract, comes in and gets into the cell through this. First it arrives at the neuromuscular junction. By the way, neuromuscular junction, let me introduce you, is a connection between muscle and nerve. Neuromuscular junction is a connection between a nerve and a muscle. So when impulse which comes from the nervous system arrives at the membrane of a muscle cell, they flow into the T-tubule. This T-tubule is connected to the sarcolemma here. By the way, this part goes out and is connected to here. On this sarcolemma, electrical impulse which came from the nervous system distributes, and when it distributes, it reaches here. When it reaches here, it stimulates the terminal cisternae or sarcoplasmic reticulum, in which calcium is stored. So calcium will be released from the sarcoplasmic reticulum. Why does calcium need to be released? Because you remember earlier I showed you calcium needs to be attached on the cover of the you remember here on the troponin tropomyosin for it to move and expose the active site on thin myofilament. Where does that calcium, which attaches the troponin to troponin tropomyosin, which is this yellowish string-like structure, when calcium comes and attaches itself to this, then this moves and exposes these dimple-like parts of the thin myofilament, which is active site, to which these myosin heads attach to further cause contraction. So going back to the picture where we were, which is this one, I was telling you about this triad, triple, which consists of terminal cisternae, which is part of the sarcoplasmic reticulum. You remember sarcoplasmic reticulum in other cells also. It's one of the organelles in every cell. Here is a T-tubule, which you don't find in other cells, which is connected to the sarcolemma or plasma membrane of the skeletal muscle tissue. So when electrical impulse which comes from the nervous system arrives to the sarcolemma at the neuromuscular junction between a nerve and a muscle, it reaches here, and when it reaches here, it stimulates these terminal cisternae, which are parts of the sarcoplasmic reticulum, and sarcoplasmic reticulum releases the calcium which is stored in it, and that calcium goes and attaches itself to these yellowish structures. So this line, this string-like structure which was covering the active site on the thin myofilament, moves and makes the active site available for attachment of this myosin head to it. You will see this on animated video how all this happens. For now, follow our discussion, please. Okay, here is a picture of neuromuscular junction. Do you see neuromuscular junction here? This is a nerve, and this is sarcolemma. or plasma membrane of a cell. Does this remind you of plasma membrane? Yes, it should. Do you see phospholipid bilayer? So when this is magnified, this is what you get. Here is a nerve ending, the end of a nerve, and here is a segment of the sarcolemma or plasma membrane of a muscle cell. So this is neuromuscular junction, meaning connection between a nerve ending and a skeletal muscle cell plasma membrane. Here is a picture showing neuromuscular junction. This is, by the way, a section through the spinal cord. This is a section of a spinal cord. Look at a nerve coming and getting attached to a skeletal muscle cell or a muscle fiber. Look at this. Look at this junction. So each and every skeletal muscle cell is attached to a specific nerve or branch of a nerve. This is a nerve, by the way, coming from the spinal cord and attached. A muscle which has lost its connection to the nervous system is paralyzed, won't function. I was telling you about electrical impulse coming from the brain or spinal cord and stimulating the muscle cell to contract comes through these lines. Look, this is real picture taken from a muscle. So for a muscle to contract, it must be stimulated by the nervous system. Skeletal muscle must be stimulated by the nervous system, either brain or spinal cord, for it to contract. So that impulse, stimulating impulse, comes through this nerve which is connected. So this is neuromuscular junction. Let me show you a more detailed picture of neuromuscular junction. Here is a neuromuscular junction. Here is a skeletal muscle cell. Here is a neuromuscular junction. By the way, when the impulse, which is represented here with red arrow, arrives here at the neuromuscular junction, neuromuscular junction is a junction between a nerve and muscle cell at the sarcolemma. By the way, this part of the sarcolemma or part of the plasma membrane of the muscle cell, which is attached to the nerve ending, is part of the neuromuscular junction. It is part of the neuromuscular junction, and this part of the sarcolemma is known as motor end plate. This is nerve ending. So motor end plate is part of the sarcolemma, part of the plasma membrane of the skeletal muscle cell or skeletal muscle fiber, which is attached to the nerve ending. So when this impulse represented by red arrow arrives here, it is transferred onto the sarcolemma and it moves along all over the skeletal muscle fiber and it arrives at the triad. You remember the triad I showed you? This is the transverse tubule, this is the terminal cisternae, which is part of the sarcoplasmic reticulum, and I told you that sarcoplasmic reticulum stores calcium. So when the impulse arrives, that opens the gateways for calcium to come out. Do you see these red particles are calcium? Calcium comes and calcium attaches itself. I am magnifying this picture. Calcium and comes and attaches itself to the troponin tropomyosin, or that yellow string on the thin myofilament, and attachment of calcium to that thin string, which is troponin tropomyosin, moves the string or the troponin tropomyosin and exposes the active site on the thin myofilament or actin. Now active site, the dimples, are exposed. They are available for myosin head to attach to it. Look, do you see? And then the myosin head strikes and moves the thin myofilament. The next thing I want us to see is the Next thing I want us to see is the steps of muscle contraction. Look here. Here are steps of muscle contraction. For muscle to contract, two things are needed. One is, look, the calcium attached to the troponin, tropomyosin, which was covering these active sites, these dimples. Because calcium, these two pink particles, which are calcium, are attached to the troponin tropomyosin or yellow string, this active site is exposed, being available for myosin head. The second thing needed is ATP. ATP is the energy, source of energy, power for the muscle contraction to happen. So when ATP is here, and when that calcium removes the cover and the active site is exposed, this myosin head cocks up, gets ready, and at the same time ATP is dissociated into ADP and PI. By the way, ATP is abbreviation for adenosine triphosphate. So adenosine triphosphate is broken down into adenosine diphosphate and phosphate. So when this breakdown happens, energy is released, and this myosin head uses that energy and it unbends. Look how bent this one is. Look, this one is to some extent unbent, kind of straightened to some extent, not completely. Compare this with this one. The next step is this unbent, cocked up myosin head attached to the active site on the thin myofilament. This is known as cross bridge formation. This is ATP being attached to the myosin head. This is ATP being dissociated into ADP and P. And then here, cross bridge formation, meaning myosin head being attached to the active site on the thin myofilament, and then the myosin head applying stroke or pulling the thin myofilament towards the M line. This is how contraction takes place. So multitude of such strokes, such pulling by the myosin head on the thin myofilament happens, and this causes muscle contraction. By the way, this can repeat if another stimulus comes after the other. This can take place again and again. So these are the 4 steps of muscle contraction. To repeat, here ATP is needed, and when ATP arrives here, what happens is, if this myosin head was attached, it gets detached. So cross bridge breaks, detachment happens, and then when ATP is dissociated into ADP and P, this myosin head kind of straightens, and the next stage is myosin head attached to the active site on the thin myofilament, and that myosin head pulls the thin myofilament towards the M line of the sarcomere. This is how contraction at the level of sarcomere takes place. This is how contraction at the level of sarcomere takes place. After you see this, after you go watching this lecture video this long, I want you to watch the animation video which you find in this lecture resource page. This, after you watch this much, go and watch the animation I have posted on this lecture resource page of module 6. Next, I want to talk to you about the methods by which our cells, our muscle cells, produce ATP. We just saw that ATP is needed for muscle contraction. So if ATP is needed for muscle contraction, muscles must make their own ATP. And muscle cells make ATP in 3 ways. This table shows you 1, 2, and 3 methods by which muscle cells make ATP. One of them is the direct phosphorylation or they also call it the single enzyme method. That is, creatinine phosphate is used to attach phosphate to adenosine diphosphate. Do you remember we saw earlier just now that when ATP is used, ATP is dissociated into ADP and P, right? So this is reversing that process. It brings and attaches phosphate with ADP, producing ATP and creatinine being released as a byproduct. So this is single enzyme method of ATP production. That's one of them. And this is the fastest. The next method is glycolysis. Glycolysis is breakdown of glucose during which ATP is formed and pyruvic acid is produced as a byproduct. This byproduct, pyruvic acid, is further transferred into the mitochondria. You know from our earlier study of cells that the mitochondria is the powerhouse of a cell. Mitochondria uses the pyruvic acid to produce further more ATP. Look how many ATP, 32 ATP is produced. So 3 methods. This is the fastest, this is the slowest, but the most productive. Look, 32 ATP per glucose molecule. Only 2 ATP per glucose molecule. In this method, which is known as anaerobic or glycolysis, oxygen is not used. Did you see crossed out? But here oxygen is used. So they call this aerobic, a process which happens with the use of oxygen or air, and this one anaerobic, in which oxygen or air is not involved. So single enzyme method, where creatinine phosphate converts ADP into ATP, one only made per process. Here only 2 ATP per process. Here so many, 32 ATP per glucose, which is a continuation. But if only glycolytic method is used to produce ATP in a muscle, then this pyruvic acid is converted into lactic acid, and that's what causes SOR, by the way. So this is the fastest, this is the slowest, but most productive, producing so many, and this is intermediate, as you can see. So these are the 3 methods by which skeletal muscle cell, or any muscle cell by the way, produces ATP which it needs for energy provision purpose. Okay, let's move on. There is one more concept I want to introduce to you, and it is, I think, here. Okay, let's talk about it. In addition to showing neuromuscular junction, this picture shows us the idea or the concept of motor unit. Do you see here motor unit 1 and motor unit 2? By the way, this picture, as I said, shows the neuromuscular junction, meaning junction or connection between nerve and muscle cell. But in addition to that, it also introduces the idea of motor unit, the idea of motor unit. Motor unit is a nerve, this nerve, one nerve which comes out of the spinal cord, and the muscle cells to which that nerve is attached. For example, the pink one is attached to 1, 2, 3 skeletal muscle cells, whereas this violet one is attached to 2 only. So this motor unit number 2 consists of 1 nerve and 3 skeletal muscle cells, whereas this one consists of 1 nerve and 2 skeletal muscle cells. Motor unit 1 consists of 1 nerve and 2 skeletal muscle cells. So every time impulse or electrical impulse comes along this nerve, these 2 skeletal muscle cells are stimulated together, they contract together. They are a team, a unit, which function always together. So the less the number of skeletal muscle cells, attached to one motor neuron, the more precise the action is. Look, the reason why our hand is more capable of doing precise activities than our feet is because the number of skeletal muscle cells per motor unit in our hand is much less number of muscle cells are in one motor unit in our hand than in our feet. That gives our hand more dexterity than our feet. Even if you don't understand why we have more dexterity on our hand than on our feet, all you need to know about motor unit is one nerve and the muscle cells to which that nerve is attached. In this case, motor unit 1 consists of one nerve and two skeletal muscle cells, whereas motor unit 2 consists of one nerve, the pink one, and three skeletal muscle cells. This is the idea of motor unit. So this motor unit always functions together, because impulse which comes along this will reach all three and they will contract always together. Here is another concept. Look, what I want you to notice here is that this muscle to which 3 kilograms of weight is attached contracts and gets shorter and lifts the weight together with it. So when this muscle gets shorter, the contraction is known as concentric contraction. Concentric muscle contraction, meaning getting shorter. Whereas in this one, it's not getting shorter because the weight is 6 kilograms, double that. If it is the same muscle, maybe it is too heavy for the muscle to lift it. So if it doesn't shorten, if it doesn't get shorter, you call it isometric contraction. Why isometric? Iso means the same. We have come across this prefix before. So the length of the muscle remain the same. Because of that, you call this isometric contraction. But this one, you call it concentric. But if the muscle was lengthened, if it got longer than it was, you could call it eccentric. If it gets shorter, concentric. If it gets longer, eccentric. If it remains the same, isometric. So when a muscle tries to contract, if it is strong enough and gets shorter, you call that contraction concentric. Isotonic because the same tone. Whereas if it remains the same, isometric. But if it gets longer, then you call that eccentric. It gets longer when the weight is too heavy. That's when people say, I pulled my muscle. You might have heard people saying when I was moving a house, changing a house, and was carrying furniture, and I pulled my muscle, meaning their muscle was attempting to lift very heavy weight, too heavy for the muscle, so the muscle got longer, it gets injured. So that was eccentric contraction. If a muscle is pulled and overstretched than it should, eccentric contraction. If it gets shorter, concentric contraction like this one. If it remains the same, isometric. These are the three choices which can happen to a muscle when it tries to contract together with it, or against a resistance, or trying to lift weight. Those are three types of muscle contraction. Muscles are classified into three groups on the basis of three criteria. The criteria are how quickly the muscle contracts is one criteria. The other criteria is how does that muscle cell produces ATP? Does it produce predominantly by glycolysis or by oxidative method in the mitochondria? So I'm saying muscle fibers or skeletal muscle cells in our body are divided into three groups on the basis of two criteria. The first criteria is how quickly the muscle contracts. Second criteria, which method does the muscle cell use to make its ATP? Does it predominantly make its ATP by glycolytic method or by oxidative method, which happens in the mitochondria? So the three groups are slow oxidative. What does it mean? Contracts slowly but produces its ATP predominantly by oxidative method in the mitochondria, mainly. It doesn't mean it doesn't produce by glycolytic method. It also produces by glycolytic method, but mainly most of the ATP comes from oxidative method. Such a muscle cell is considered fatigue resistant. Not considered, it is fatigue resistant, meaning it doesn't get fatigued easily. So people who have most of their muscle of this type, if a person has most of his or her muscle of this type, slow oxidative type, they are good for exercises or sports which need endurance, like marathon, where you have to be running for a long distance for an extensive period of time. So they are fatigue resistant because they produce their ATP by oxidative method, so no accumulation of lactic acid, because oxidative method completely decomposes glucose. And the second method is opposite one, is fast glycolytic. This is contracts quickly, but it produces by glycolytic method. So the people who have this, they quickly get fatigued, but they can quickly produce high power. For example, weightlifters or sprinters which run only 100 meters. Those people who have predominantly this type of muscle cell which contracts quickly but predominantly produces its ATP by glycolytic method. If a person has predominantly this type of muscle cell, they are good for weightlifting or sprinter running, where they need to produce high amount of energy within a very short period of time, but they get fatigued quickly. These ones are in between. By the way, your textbook calls this intermediate, meaning they are in between the two. They are not that slow, they are not that fast. They make their ATP by both methods, by the glycolytic method and an aerobic method. So these are in between. So such people may not be very good sprinters, or they may not be very good at endurance requiring exercise. They will be in between, but through exercise these muscle cells have a chance to be changed into this one, into slow oxidative one. That's what shows. So what does exercise do to our skeletal muscle cells? Exercise can cause our skeletal muscle cells, can make them hypertrophic, meaning getting bigger and stronger depending what type of exercise we do. If the exercise we do is like weightlifting, which is quickly producing high tension, then it can cause hypertrophy. Hypertrophy means increasing in size, whereas aerobic exercise makes our muscles stronger, meaning resistant to fatigue. That's what exercise does to our muscle. If we do an aerobic exercise, meaning short-term, high energy, high tension producing exercise, that will increase the size of our muscle. Whereas if we do aerobic exercise, which is long-term but lightweight exercise, that increases endurance to our muscle, meaning our muscle will not get fatigued quickly. That's what our muscle can do to our what exercise can do to our muscle.